start. That's like welcome, good afternoon, and welcome to the virtual biodiversity data use workshop. So let me share my screen and let's start today's session. Okay. So actually, we had the first round data use workshop in March, and we decided to have the second round this week because we received more than 100 applicants and we can only provide the limited seats. So that's why we decided to have the second round and invite all of you who didn't who were not able to join last time. So before the session starts, and let me introduce myself and I'll let my partners introduce themselves as well. I'm Melissa Liu from Taiwan, together with Lily Stresa and Vijay Barve from the GBIF Asia Regional Support Team. I have also been working at the GBIF node in Taiwan since 2018, so also known as Thai BIF. And we will be leading you through the next 2 days, today and tomorrow, on how to use GBIF mediated data for biodiversity research. And we hope you can learn from the great resources as we delve into the course content. So I'll let maybe VJ introduce yourself first.
Hello, I'm Vijay Barve. I'm from Nashik, India. I've been working on biodiversity informatics and GBIF data for a very long time, and I'm happy to be here with you all to share how to use GBIF data. So I'm looking forward to interactions over the next 2 days. Over to Lily.
Hi everyone, good afternoon. My name is Lily Shrestha and I'm from Nepal. So I've also been working with my colleagues here, Melissa and Vijay, in GBIF Asia support team for last 4 years. And the primary work that we do in this role is to help the data publishers, the data holding institutions in Asia publish their data, or anyone who's interested in using the data. So that's basically the technical support and help desk role that we do for the Asia region for GBIF. I'm just involved in this role for now. I look forward to working with you in the next 2 days. Thank you. Nice to see you, everyone.
Okay, thanks VJ and Lily. And then we go to the next part. Okay. Before we introduce the activity of these 2 days, I'd like to cover some housekeeping and ground rules. The first is the Zoom session will be recorded and they will be available for all the participants after a workshop. So when you sign up for the workshop, we ask you to complete a media release. If you did not agree to the media release, then you should please keep your camera off so that we are not recording your image. And if you post on social media, please use the hashtag GBIF Asia and tag GBIF. Do not share any photos of people and screenshots of materials or presentation is fine. We will do Zoom selfies today, later, and tomorrow, and that photo can be shared as people who don't want their image shared can opt out. So it's good to keep your microphone muted to minimize background noise during full group sessions. You can use the chat function below the toolbar and the chat box to ask questions or contribute additional information. You can also use the reaction button to raise your hand if you like to be called during discussions. But if you cannot find the reaction button, just raise your hand and turn on your camera. Maybe we will see you. Okay. Today we will not have breakout sessions this time, and we'll mostly discuss all the exercises in the main room, and tomorrow also. So please unmute your microphone and participate during the discussion sessions. And this is your time to ask questions. Okay, so we open for all the session. And if you need to miss a session or part of a session, please also let us know in advance. Okay, you can message in chat box or email us. Okay, and today we will have basically two sections. The first section will be the introduction to GBIF, and okay, and then sorry, I didn't change change the agenda yet. And then we will go to using GBIF mediated data. And then we will have the first exercise, which is Darwin Core. And then we will have gbif.org search functionally presentation and go to the exercise 2, gbif.org exploring. Then we will have a group photo selfie and have 15 minutes break. And then the section 2, we will introduce about data quality issues and flags. And we will have the exercise 3, which is examining download issue flags. And we will have QA for all sessions. Then tomorrow, day 2, we will also have two sections. Section 3 will be about the GBIF API. And we'll have exercise 4, text some keys in Q&A and introduction to RGBIF. So I hope all of you already installed RStudio before tomorrow's sessions. Okay. And then again, tomorrow we will have group photo selfie and 15 minutes break. And then the last section would be the R demonstration. So we will let you follow the instruction to use our GBIF package, and we will have discussion and open Q&A. Okay. So let's start with the first section, and I'll give the floor to Lily to introduce to GBIF.
Sure. Thank you, Melissa. Hi, everyone. Again, let me start by sharing my screen. Can you see my slides?
Yes.
Okay. So in this first part of the presentation, I will be giving a quick review of what you have learned. If you remember, I've sent an email saying you need to go through the Introduction to GBIF course, everything about what GBIF is and what it does. So this couple of slides in the beginning will just be a review of what you have learned there. And then we'll also be talking about aspects of using GBIF mediated data, what things should be considered when we are using GBIF mediated data. So these are the two parts of the presentation that I will be talking about in this next 20-25 minutes. Let me know if my slide is changing. Yes? Okay, so starting with what GBIF is, we all know GBIF stands for Global Biodiversity Information Facility, and it is an international network and data infrastructure that is funded by the countries that are participant to GBIF. So GBIF exists aiming to provide anyone, anywhere, free and open access to biodiversity data. But nowadays, because there are a lot of records, as you can see in the screenshot, more than 3.6 billion occurrence records, GBIF is also tracking the use of GBIF mediated data, and we are also encouraging people to use the data that's already been available through GBIF. GBIF works with the participant countries and organizations through a memorandum of understanding that is agreed between the two, and basically it's a voluntary collaboration. The core team of GBIF is located in Copenhagen, Denmark. That's called the GBIF Secretariat. So the screenshot here you can see is the homepage of gbif.org, and we have a dedicated exercise on exploring gbif.org, so you will be seeing this later on as well. I will not explain much about it, but we can just note the numbers in the bottom that displays the richness of biodiversity data that's been made available in GBIF, like more than 3.6 billion occurrence records that has been published in the form of more than about 122 data sets by 2,686 publishing institutions from all around the world. And as I said, GBIF is also tracking the use of GBIF mediated data. There are up to now, there are 14,603 peer-reviewed papers that have found to be using GBIF mediated data. Next slide is on the occurrence records in GBIF that has multimedia evidence. So we can see there are 267.4 million records out of all the 3.6 billion occurrence records that has taxonomically identified images link associated with it. And additionally, 1.8 million occurrence records had audio files, and 5,478 occurrences has videos associated with the occurrences. So this type of records that has multimedia evidence associated with it can be very useful when we are looking into the evidence-based conservation action or decision making of biodiversity. The next slide is on the network of data publishing institutions from all around the world. So there are 148 countries or areas that have institutions that are publisher to GBIF and have started sharing data through GBIF. The top being United States, United Kingdom, and so on. You can see in the graph, and the color coded in the map also shows the publishing institutions from all around the world. This diagram here generally shows how the occurrences or observations, species observation comes into GBIF, how it is shared, and how it is made available for use. Starting from the left-hand side of the diagram, the source of data in GBIF are from these institutions, including the Natural History Museum, government agencies and ministries, field science NGOs, INGOs, citizen science networks, a business organization who usually does environmental impact assessment and records species, and university, obviously academics is a huge part of it as well. They record the observation of species either in the form of specimens, mostly in the Natural History Museum, field observations. Sometimes the observation also comes through the literatures that has already been published. Also sensor based, because most of the records also comes from, for example, camera trap or remote sensing. And more commonly these days, environmental DNA sampling that also provides the occurrence of species in nature without actual visualization of the species. So these are the source of data that comes into GBIF. Once they have the data, they need to standardize it, and in GBIF we use Darwin Core standard. I'll also be talking about it a bit later on. And the data needs to be shared using open licenses. I will also be talking about the license in one of my slide later on. Once the data set is ready, it should be uploaded. It not should, but the most common way of publishing the data in GBIF is through the integrated publishing toolkit. That is the software that through which we can publish our data. And once we have published and registered, GBIF simply indexes the records that is in your dataset and makes it available through cloud services, gbif.org, and API. So API gbif.org is something we'll be covering over the next 2 days with a practical exercise as well. And then once the data is available, you can use for your own research purpose or policy making around it. Next, I would like to quickly summarize the type of data set that you have already, I think, reviewed in GBIF, in Introduction to GBIF course. There are 4 levels of data that is supported in GBIF so far. The first one being dataset metadata. It is just a description of your dataset. So whether it be taxonomic, geographic, or temporal scope of the dataset, but it does not contain actual record of the species. This is the only type of data that is different from rest of the 3 dataset that is supported by GBIF. The next one is species checklist. as the name says it, it's a checklist, list of taxa within any region or thematic area. So it is just a list of species and does not contain other information, including geographic or any other information, just about the taxonomic and nomenclature status. Third, and the most common type of dataset is the occurrence-only dataset. So in occurrence-only dataset, obviously we will also have the record of species, list of species, but along with that, we'll have the date of observation, when the species was observed and where the species was observed. So the geographic locality information such as coordinates. That's what occurrence dataset is about, and that's the main source of all the occurrences you see in GBIF. And lastly, the most complex form of dataset in all these four is the sampling event dataset. In sampling event dataset, why is it complex? Because it will also have the occurrence data, but in addition to that, we'll have a separate file that will contain all the information about the sampling methodology that was used to, gather the observation of species in the field. So sampling event dataset is about that. As I mentioned in my slide before, we use a standard in GBIF, and that's called Darwin Core Standard. Darwin Core is just a list of fields and their definitions relating to biodiversity data. Why we use standard in GBIF is because I've already mentioned there are like 148 publishing institutions from all around the world that has been publishing data in GBIF. So there's a lot of varied sources and countries that has been sharing data in GBIF, and therefore we need a standard to have a common understanding of the data that is being shared. Otherwise, the data could be misinterpreted. So that is why we use Darwin Core Standard, and we have a very useful resource that helps us that helps us in identifying what kind of terms can be used, what kind of data can be included in those terms. That's called Darwin Core Quick Reference Guide. Okay, so the next slide is on GBIF tracking of using GBIF mediated data. As you can see in the slide here, the GBIF has been tracking data from use of data from 2008. In the year 2008, there were only 52 peer-reviewed publications using GBIF mediated data, and that has steadily increased over the years, reaching to 2,254 in 2024. And in 2025 it's the data until 1 November 2025, so it's around 2218. Obviously it should have increased, but GBIF has not provided this data yet. So we can see that not only publishing of data but use of data has also increased over the years. So this was all about quick introduction to GBIF based on the course that you have taken. Now the next part of my presentation, I will be talking about using GBIF mediated data. So let's start with saying that the data used in GBIF is interdisciplinary, and this is an outcome of a systematic review of GBIF use of GBIF mediated data by Heverling et al. 2020. So in this study, a comprehensive analysis and review was done of more than 4,000 peer-reviewed studies published between 2003 and 2019 that relied on data from the GBIF network. And this particular study showed that both data availability and data use have increased over time, which GBIF's tracking system has also shown, as I just talked about it earlier. And it also showed that uses of GBIF media data span disciplinary boundaries and are conceptually diverse, as we can also see in this figure here, that the GBIF media data is not only used further in the field of biology, but also in the field of health science, chemistry, biotechnology, social sciences, and so on. Let me also talk quickly about the relevance of GBIF mediated data in addressing the global policy needs, such as the Kunming-Montreal Global Biodiversity Framework, which is the most important framework to look at when we are concerned about biodiversity. So we can see GBIF, the arrow shows the relevance of GBIF's data to the target of global biodiversity framework. It is related in many different ways. For instance, when we look at target 1 to 8, set for reducing threats to biodiversity, GBIF provides primary data on species occurrences, which is required for target 1 on identifying key biodiversity areas, target 2 on monitoring restoration of ecosystem, target 3 on protected areas, target 4 on species conservation, target 6 on invasive species management, and target 8 on reducing climate change impacts. Similarly, when we look at target 15 on assessing risks from business, GBIF does provide platform for sharing EIA data. And lastly, the targets that are directly related to what GBIF does is target 20 on capacity building for biodiversity, and that is what we are doing today as well. We provide training workshops on data use, data mobilization based on GBIF biodiversity data infrastructure. And lastly, to target 21 for making best data available. As I've already mentioned, GBIF exists to make species data freely and openly available for evidence-based conservation action and decision making. So this is how GBIF is related to the Kunming-Montreal Global Biodiversity Framework. Another Sorry, there was a slide in between, I guess. Yeah. So another big global policy framework to look at when concerning about biodiversity is also the sustainable goals. And GBIF supports quite a broad range of SDGs. When we look at its relevance to SDGs, the most easy to look at and are directly related to is obviously SDG 14 and 15 on life below water and life on land. GBIF does provide occurrence records for the conservation of life below water and life on land. Other SDG that GBIF mediated data could contribute to are SDG 2 on zero hunger by providing data on agricultural species, fisheries, and many aspects of food production. SDG 3, good health and well-being. The data available through GBIF supports policy goals related to human health, especially with regards to disease vector. And lastly, in relation to SDG 13 climate action, GBIF supports global effort both to mitigate and adapt to climate change. Example, modeling species range shifts due to climate change. So this is how GBIF could also be linked to the SDG. One of the example where GBIF mediated data has been used widely is the ecological niche modeling. We all know ecological niche modeling is a study where we predict suitable habitat for species based on the data of where the species has currently been found. This model correlates the occurrence data of species from their native range, and one of the source of that can be GBIF with environmental data provided at a given resolution. Species occurrence data should actually reflect the precision of environmental data used. So higher the number of data points, species occurrence points, the better the model fit. So GBIF can be a very important source of this occurrence data of species that we are concerned about. Next example where data has been used, GBIF data has been used is the red list assessment of IUCN, which assess the extinction risks of a species against a set of objective criteria. And one of the important criteria is criterion B on restricted geographic range. For calculating restricted geographic range, there are two metrics, extent of occurrence and area of occupancy, both of which require georeferenced locality data for which GBIF mediated data can be a very good source. GBIF occurrence points can also be used to produce species distribution map that are required to accompany the assessment of IUCN. These occurrence data used in the assessment evaluate whether species meet geographic range size threshold and that indicate whether the species are in extinction risk or not. So these were the few examples how GBIF could be related to, global policy framework, or how GBIF data could be used in some of the studies or some of the important assessment like IUCN. Now let's move into the access point of GBIF. So by the ways by which you can access GBIF mediated data, and there are most commonly three access point. The most common and easy access to the data is through the gbif.org website for people who are not into programming, and this actually offers advanced searching, data analytics, visualization, and you can also download the data. For people who are registered, so one must have an account. We'll also be talking about that later on. And GBIF data or gbif.org is now available in 12 different languages. Next is a more programmatic way of accessing the data by using GBIF API. API can be used to make advanced queries that are not supported by the website. And one of the ways of accessing GBIF data through API is R, for instance. And R users can use GBIF, access GBIF mediated data using R package that's called RGBIF. We will be learning more about this and also doing a dedicated exercise with R scripts in tomorrow's session that our colleague Vijay will be leading. So let me talk quickly about the golden rules of GBIF using GBIF mediated data, what you should consider when you're using. So these are the 5 points I'll also be discussing in the coming slide. So let's not focus on that now. The first one is you need to have an account in gbif.org. That is what I meant by saying registered user. So you need to register in GBIF in order to access the data that's available through GBIF. Obviously, you can explore the website without the account but for downloading, you do need an account. Not only for downloads, but you can also look at the history of your download that you have done before if you have the account. And any derived data set that you may also have. So you should always use a dedicated email and password pair, as it is also most straightforward in the programmatic ways of accessing GBIF data. I have mentioned that GBIF exists to provide free and open biodiversity data, and obviously GBIF data is free to use, but it's not free of responsibilities. Every user must agree to the GBIF data user agreement, which is actually a non-binding agreement between the users and GBIF. It sets out the rule by which users should be using data, including due attribution to the use of the data through correct citation practices, attributing the publishers, using the data according to the license with which it is made available by the publisher. Again, I'll be talking about license just in the coming slide. And also notifying GBIF regarding any data quality and other issues concerning their website or data availability using the feedback mechanism provided by GBIF Secretariat. So before using GBIF data, please carefully read the principles set out in the data user agreement. I've talked about license a couple of times, so this is what I meant. That every record in GBIF is shared using along with one of these three Creative Commons license, starting with the most open one, that is CCO, that has made data available for any use without any restriction. CC BY, making data available for use, but it says that the users have to give appropriate attribution to the publisher. And lastly, CC BY and C somewhat restricted than other two licenses because it does not allow for the use of data for commercial use. And it also says that appropriate attribution should be given to the publisher. So these are the three licenses. Every record in GBIF is associated with it. So we should be very careful about using data when we are using data, and check their licenses accordingly. So GBIF data is not free of issues. It obviously has some issues associated. So we will also have a dedicated session afterwards on identifying issues and flags and how we can think about that when we are post-processing the data. So obviously some post-processing of the data is required, and this obviously can happen with the filters that you apply in while you are downloading in gbif.org, or you can also process it through programming languages such as R. Processing data will always require an approach that will balance the requirement of your analysis, that is, in terms of level of precision, for example, and the robustness of your analysis. So there's no right or wrong way to process your data, but you should make decisions on what you keep and what you don't keep based on what your study demands. I've already mentioned we'll be talking about the issues and flags that might be associated with the data you are looking in GBIF. And after we download the data, we should always document the process by which we have we have post-processed the data, because the data in GBIF is always changing, as you know. Once you have done some kind of analysis today and you want to repeat the similar kind of analysis in future, the data in GBIF might change. So you might need to repeat the process again, and that's when the documentation of process, how you have processed your data, could be very useful. So after you have done all processing and you are ready to download your data, you have your account set up, data can be downloaded in 4 formats. Starting from a simple one, this is a much smaller file that contains a smaller number of only GBIF interpreted fields, no raw data and no multimedia if there is any within the data set. The second one, which has everything, is the Darwin Core Archive. This contains all of the original data as well as the interpretations from GBIF, and also includes link to multimedia if the user publishers has provided. Species list, it is just a list of species names found within the data set and won't have anything else. So it is just the interpreted data. And lastly, a relatively new addition is the cube download. This download format allows you to aggregate the occurrences that you have selected by their taxonomic, temporal, and or spatial properties. So it can be very useful when you are doing modeling, and the result is a CSV file. Lastly, it is very important that you correctly cite the data that you have used. Every data downloads are issued with digital object identifiers, as we call it DOI, that users are encouraged to use when they cite the use of data. GBIF's literature tracking can detect the use of DOI from Google Scholar, Scopus, Wiley Online Library, Springer Link, NCI PubMed, and so on, that allows GBIF to show how data provided by data publisher is being used. And obviously, citation practice varies depending on how you're using GBIF, and you can get a full overview of the correct citation practice from the first link here on citation guidelines. And a new development in GBIF is derived dataset tool, which allows users to retrieve a new DOI only on the data that they have actually used. Sometimes what happens is we download a couple of data set and we process it and we use only the fraction of it by combining different occurrences from different data sets. So if we want a separate DOI for the type of data that we are using, we can use this tool. We can let GBIF know which data set we have used, which records we have used, and they will provide us, GBIF will provide you a different DOI to be used for your study. Yes, and lastly, once you have processed and have used your data for analysis, please deposit in your processed data in a public repository such as Genodo with the associated download DOI, just to facilitate access to your data in the future. Just summarizing the things that we need to consider, the principles that need to be considered when we're using GBIF mediated data. You should have an account on gbif.org. You should agree to data user agreement. It is always recommended to document how you have processed your data. You must correctly cite the data that you have used, and it is also recommended to deposit the used data in a public repository. So this is all my presentation was. Thank you for your patience and listening. So I think our discussion will be towards the end, right, Melissa? Over to you. Thank you, everyone.
Okay, thanks, Lily. And then we just like go to the first exercise. Let me share my presentation. Okay, I hope you can see my presentation well. Okay, and so the first exercise is exploring Darwin Core. In case some of you might be the first time who like hear about the Darwin Core. So let me have the basic introduction about the Darwin Core. And before like you use or publish the data to GBIF, you need to know about the data standards. Data standards are the rules by which data are described and recorded. So in order to share, exchange, and understand data, we must standardize the format as well as the meaning. So there are a couple of the biodiversity data standards. So the most important is the Darwin core, because that's the GBIF mainly used for the data structure. And so the basically is the common format of the data fields. Like for example, we need to use the same field names to know like name, species, something all is meant to the same one scientific name. And the second is to like follow the same format of the data content, especially like the date thing should be like because like for in Europe they will use the date month year, and in America will be the month date year, and other might be year month date. So we just use the same format like year month date in the same data. Okay. And all the data standard, the main purpose is to be readable for everyone and machine. Like anyone download the data from GBIF, then you can know what's the field name mean and what's the content mean. Okay. So just like what I mentioned before is the standard that GBIF is using is Darwin Core. Okay. And Lily just like introduced about the three mainly like the classes of the data set. So if we put the Darwin core terms to the different data classes, for the checklist, it will be like you will have like maybe taxon ID, scientific name, because it's the checklist for the species for the region, and scientific name like a kingdom, taxon rank, vernacular name, and other like taxon level, like phylum, class, order, family, genus. So for the occurrence data class, dataset class, should be like include occurrence ID, event date, scientific name, kingdom, taxon rank, and individual count, locality, decimal latitude, decimal longitude, country code. So for occurrence one, the location and the species should be the most important. Okay. And the time also. And for sampling event should like include event ID, event date, sampling protocol, sampling size value, sampling size unit, habitat, and sampling effort. So it's all about the information about the event. Okay. So that was the common data Darwin core term that we'll use in different dataset classes. Okay. And so in the first exercise, I hope you already downloaded the exercise sheet. The scenario is that when you get your data, your spreadsheet will like contain a large number of terms that you may or may not be familiar with. The data available is like often much richer than you think. So in this exercise, okay. Sorry. In this exercise, we will introduce you to a small slice of total number of Darwin core terms available to you. So if like not sure if it's your first time to see Darwin core, then you would like to do a study using GBIF mediated data to characterize a species habitat and determine population sizes. Okay, so here are like what would you use for this kind of study? So the terms here are individual count, occurrence status, associating media, associated taxa, organism ID, material sample ID, event date, habitat, sampling protocol, minimum elevation in meters, and decimal latitude, type, status, property, and vernacular name, and recorded by. So these are the terms in this exercise you may or may not use. So in the exercise one sheet, Okay, just choose, no, just just list all the terms here. Okay, and say like if you think the terms is relevant to the study or not, and why. And I'll give you about let me think, 10 minutes, 10 to 15. Let me see. Okay, 10 minutes. And then I'll pick up someone or some volunteers to show your answers. Okay. And I'll set up the timer. Okay. I hope you already check all the Darncore terms on the exercise one sheet. And who is can be the volunteer? Please raise your hand to show your answers. If no one volunteered, I would like to pick one to just like show your answers is fine. Is doesn't matter if it's correct or not. We can like discuss together. Okay. Ouch. Let me pick up one. Um, maybe Johnny? Johnny Conjara?
Yeah. I will be sharing my screen.
Okay, thank you.
Yeah, is my screen visible?
Yes, great.
Actually, it was not completed. I'm just still, working out. So some of the things I just identified.
Yeah, you can like also like say like why you choose some of the terms and why not.
Yeah. First thing is, the listed terms individual count is, it is essential for, estimating the population size or trends. So possible quality issues are if there is any missing values or if there is any rough estimates. So we don't know exactly the provided information is accurate or not, whether what kind of counting methods they use. So this is the one of the important term, and then what are one of the quality issues we will normally will face. And then second one is occurrence status. So this is also one of the important one. So whether it is present or absent, so will comes to know. And then it doesn't mean if anyone is keeping absent, it doesn't mean that it is true absence. So mostly will be considered as a presence only data.
Yes.
And then associated media and the associated taxa. So I'm not very sure. Maybe I just kept it maybe only. So media may be useful for verifying the species whether the information accurate or not. So some of the issues are whether the if the provided one is low quality or if they identified. I mean, misidentified media means we don't know exactly the which species it just belongs to.
Mm-hmm.
And then associate taxa. So when we are having any information, if the associate taxa is there, we will comes to know the ecological interactions between the each between the species.
Mm-hmm.
And then organism ID, maybe it may be useful for to avoid the duplicate records. Possible quality issues are maybe there may be some missing IDs, or if the ID is not unique to particular data, we don't know how we can consider this one. And then material sample ID basically for the DNA occurrence information. So it is further validating the species names whether it is identified properly or not. And even date is it is must, and if it is more essential to identify the temporal trends. Habitat, I kept yes, but I didn't give in any reason. And the sampling protocol also, maybe sometimes it will be useful. And decimal latitude and longitude is important for, distributional mapping and all. And type status property vernacular name recorded by, so it may not be much useful for this kind of population size or extension of the species and all. So this is what I just prepared.
Thanks, Johnny. It's like most of the terms are like your answer is really great and correct. And I just want to add something on is like let me see. Organism ID is like if the purpose is to avoid the duplicate records, it would be better to use occurrence ID because not every dataset would provide organism ID, but every occurrence data set would provide occurrence ID. So you can like check this from the duplicates. But organism ID is mainly follow with the individuals of the animals. For example, because I'm more familiar to sea turtle data set, so I just like take this for example. Like a sea turtle, we can like use their photo for the faces to do the photo identification by individuals. So that case, we will like provide the organism ID and for each individual of the sea turtles. Then we can follow from this ID to know if it's the same one or not. So it may not be like follow with the same species, but like to the individuals. So if you want to use this term, um, just like check your target species if they can be like tracked with by individuals or not. So it's mostly like usually like mammals and reptiles or others that can be tracked by individuals.
Okay.
And others vernacular name, sometimes you may need it if your target species is like known as like more famous, known as the vernacular name. I'm not sure if you like understand the situation. It's like sometimes if it's like your data is used from the citizen science project and the public may only know about some species as vernacular name, not the scientific name. So if you like use the vernacular name, you can like do the like double check to confirm is the species you want or not.
Okay.
So like VJ or Lily, do you want to add something more?
No.
Okay.
Is
And
habitat and sampling protocol is required?
sampling protocol and habitat, yes, it's like habitat is like you can use it to know like where and when to like collect the occurrence, the data, okay, the species. So if you want to know about the population, you will need the habitat information. And the sampling protocol, depends. Let me think about. It depends on like which species is targeted, like for assessment. But I don't have any example from now to yeah. So sometimes like yeah, still depends on species. Some species may need this field, but some species may not really need. For example, like cetacean. Sorry, I'm also like marine background. So like for cetacean, like dolphins and whales, we usually use the same protocol. So if I don't even like use that term, I will know what kind of protocol they use for like collect the data. Okay, VJ.
Yeah, for botanical data, many times sampling protocol is useful because botanical sampling they put these quadrants and transects where you have a specified area and they're counting all the individuals in that and denoting that, and more data sets are being available. So as Melissa rightly said, it highly depends on what organism you're looking for. Sampling protocol may be important or may not be important.
Yeah, minimum elevation in meters that is also is required for the land fauna.
Yes. Yes, for the land fauna, it will be um helpful because you will know it's from the mountain or the others kind of habitat. But for marine mammals, for marine like species, it would not that, yeah,
Okay.
not that important. And okay. And anyone, any of you, like all the participants, do you want to share your different points of view of the terms to use? Okay, just see Anita said sometimes it gives not correct estimate about the population size, individual count. Yeah, like individual count, it's like how to say? It depends on like when you use this term, you need to be careful. Like if the count is low or high. Um, if it's low, it can like count it like it's also like helpful to refer to this field, but it's high, it's like the massive amount. Then it will not that really, how to say, not so relevant sometimes. Because you cannot analyze this from the individual count. It's like that maybe sometimes it's a group of the occurrence. And sometimes people will just provide not the numbers, but a group of it or something. Okay. And okay, and someone say, okay, Apendra said, I believe the sampling protocol, if used, keeps the good data quality. And correct me if required. Yes, sometimes it will like um be sure how like the data provider collect the data. Okay. And why only minimum elevation? No, it's not only because just that, for example, in this exercise, it just provide you this term, but you can still like, if in your case, yeah, you can like use both like minimum and maximum and to know the average or the range of the elevation this species uh like its habitat goes. Okay. Okay. And type status property. Let me check. It's interesting that, oh, I think I should also give you this one. Lily also mentioned before is the Darwin Core Quick Reference Guide, which will like uh provide you all the definitions about the Darwin Core term and the example. Okay. And about the type status property. Sorry, let me check about it. Type status. Lily or VJ, do you like are you familiar to this term?
Type status, I know, I don't know what is type status.
Yeah, I'm not
Type
sure.
status is if that particular record is describing the type, which is the specimen that was collected and used to describe.
Yeah, I'm
Yeah,
not sure.
holotype, paratype, things
Yeah,
like that.
it's about the collection thing, but I'm not sure if the type stated property because I cannot find it in darn core list.
I
Maybe it's the
yeah,
wrong
yeah, I think Ashish meant type status.
one. Mm-hmm.
Yeah, so that is correct. So uh, that is the one which is used to describe that particular species.
Oh yes, it should
Yeah.
be the type status.
Like
Okay.
holotype or paratype.
Yeah.
Yes.
Yeah.
So it's like if you want to use collection data, this will be meaningful to the study. Okay. And I think because of the time limited, we need to go to exercise two. Should be exercise two is, sorry, I lose my agenda here. Um, okay, the exercise two is about the gbif.org. So I'll give the floor to Lily.
Thank you, Melissa. So I would like to share my screen again. So in this first part, what I will be doing in this demo is that I'll be taking you through how you can explore species space on gbif.org, how you can explore the occurrences and apply some filter according to your research need, and how you can also apply spatial search for species concerned in a particular location. So that's what I will be quickly showing the demo, and then we'll be moving on to doing an exercise based on what I have shown you quickly. So this is the homepage of gbif.org. The background has currently been updated, but you can see there are different tabs on occurrences. If you would like to see the occurrences that are published, you go from here. If you'd like to see the species page, you go from here. If you like to explore the data sets that has been published in gbif, it's here. If you want to know more about the publishers that have published this data set, and if you want to access the resources or any documents that gbif has provided. So let me start because I'll be dealing first with the species space search. I'll do a free text search, and I'll search for Asian elephant that is native to Southeast Asia and Indian subcontinent, as we know. So when I do a free text search, it will give me everything that the word is in data set, publishers, species, resources, and so on. Sorry, I just forgot to mention because I was planning to mention this in the beginning when there was this homepage. gbif.org is changing soon, the interface of gbif.org, and the new interface will come live maybe few months later, but I will also show you in the end how it differs. But because now this current website is working, we have to go through this. Because now I'm looking for a species. So let's just click on the species tab. Obviously, I could just go to the species tab and do this search for sure in the beginning. My search is refined to 11 results, and if I'm still unsure which species space am I looking for, I can apply some filters here, saying I'm looking for a species, I'm looking for an accepted name, and I'm looking for a mammal. Right, so it shows me 3 results now. Obviously, I'm not looking for carnivores, I'm not looking for macaques, I'm looking for an elephant. So this is the result, that this is the page I'm looking for. Elephas maximus, with its scientific name authorship, it says that the species is accepted. It also gives the source in which the name was first published in Systema Nature 10th edition, and also gives the common name of this species in English. It also says there is a trade restrictions. We know that in CITES it falls under Appendix 1. So in left-hand side, you can see there is information about the higher taxonomy of the species, right from kingdom to subspecies. And when we explore the species space further, we can see there are 12,243 occurrences that is associated with this species currently, and there are 4 subspecies with this particular name in under this name. It's also given here. If you click on 4 intraspecies, you will have individual link and individual pages for the subspecies. Moving on, we can see that there are 5,698 georeference records, meaning the record with coordinates are only around 5,600 among 12,243 occurrences. There is another section on description where it includes description about the taxa, that could be conservation, habitat, ecology, activity, and so on, with all individual sources given. So you can refer to that as well. If you are reading the description section and taking some information from there, you can obviously go to the sources. Next section is the IUCN status, where it says that the species is endangered, threatened with extinction in endangered status, endangered criteria. Next is the vernacular name, so many different common names in English and other languages as well, German, Spanish, and more. Next section shows us where the names is found in the data set of GBIF. It says that it appears in 32 checklist datasets and in 113 occurrence dataset. So if you want to explore the dataset, you can just go here, view the occurrences, and the occurrences of Asian elephant within that particular dataset will be shown to you. And lastly, if you're using any information from the species space, the citation section, you can take the citation from here, or you can also see the guidelines and decide accordingly. But remember, citation is very important, as I've already mentioned before also. So this is how generally a species space will look like. This particular species also has another section that's called treatment. So it has all the taxonomic history and description of this species associated with this species with individual links. You can go and have a look, but remember, it's not available for every species that is in GBIF backbone. So this was first a quick demo on how a species space looks like in GBIF. Now we will go into looking at the occurrences, occurrence search, and applying some filters there. We'll do a demo of this species itself, so I can just click on this 12,243 occurrences. But the most usual way would be to go from gbif.org itself and then the occurrence, right? And this is how currently occurrence interface is looking like. It will change drastically. It will be coming another interface that's coming. You can see there are filters that you can apply here. So let's apply the filter on scientific name saying Elephas maximus that I want to look into. Same result, right? 12,243 results. Let me quickly also take you through some of the filters that is available in simple filters, and more advanced filters are available in all filters, depending on what data publisher has provided in their data set. GBIF always automatically applies this filter on occurrence status as present, because most of the studies usually demands the presence point of the species for sure. But please remember, there is also absent records that can be published in GBIF and is available. So if you want that, you can filter accordingly. Next is the license. I've already talked in detail about the license, 3 types of license that the data, every record in GBIF is associated with. So for instance, if you are doing study for commercial use, then you should just avoid this CC BY NC records 4,541. Right? So that's how you can use license. Next term that I want to quickly talk about, and is very important because it's a mandatory term for an occurrence data set, is the basis of record. So basis of record generally means how your observation was recorded. What is the basis of record for your observation? It is a controlled vocabulary, so you cannot just provide any field here and you have to choose from the set of field given. General fields are observation and occurrence, depending if you're not sure of what basis of record should apply to your records. But more specific, you can be on the basis of record. For instance, if you are recording the species from camera trap and other machines, then you can say it's a machine observation. If you're directly observing the species in field, you can say human observation. If you have some parts of the organism, or if you have a DNA of the organism, you can say material sample. If the occurrence of the organism is derived from the literature that has already been published, some books or journal article, then it will be a material citation. Preserved specimen, if the occurrence comes from the natural history collection specimens. And if it is from fossils, it will be a fossil specimen. And lastly, living specimen, when the organism is living in a zoo, or it's in a botanical garden. So if botanical garden and zoo is concerned, we provide living specimens. So this is the quick summary of what basis of record is about, and you can definitely choose according to your research need. Obviously, we can also know more about basis of record in Darwin Core Quick Reference Guide that Melissa has already provided link on. Then next is the temporal scope of the data. You can filter, you can say, for instance, if you want data from 2000 to 2026, it will filter for you. So you can just add that filter according to your need, because temporal scope is also something important when we are when we're trying to access data occurrence records. Month, you can do, when you filter the year, you can filter month as well. So spatial filter, location, administrative area, and country or area is something I will just describe later on. But let me quickly go through this section on continent. You can say you just want occurrence records from Asia. You can do that here. Data set, if you want to see occurrences in a specific data set, then you can choose and it will only show the occurrences. So that's how simply a filter is applied. If you want to know occurrences of Asian elephant published by a certain publisher, you can see here. And lastly, the very important one is the issues and flags, because as I said, GBIF's data is not like ready to be used. You have to do some processing. There might be some issues and flags associated with it, because it depends on how data publisher has provided the data. So you should always look into the issues and flags that is being flagged here by GBIF on the data that you have filtered, that you have refined. So different data quality issues. Something that you cannot do in current interface is, for all of these 12,000 something records, I cannot just say I don't want occurrences with which says country invalid. Yeah, so 29 records that says country invalid, I don't want these records, but I cannot do that in the current interface. I can only select the 29 occurrences that has country invalid, and then have to do it manually in my post processing of the data set. That's something possible, but it's not possible to exclude this in the current setting. It will be available in new gbif.org, and that's a significant improvement. I will also show it later on. But we have a dedicated session afterwards that Melissa will be leading us on identifying issues and flags and what these issues and flags means. So this is the general filter. If any filter is not available, you can just go on all filters and search if that's there. Then let's move into the spatial search, how we can apply spatial filters on the occurrences. Starting from the location, we can always say I only want occurrences with coordinates. That will only give me occurrences with coordinates, or without coordinates also, or no preferences. So let's just put no preferences for now. In this little map here, it's possible for you to quickly delineate any area and refine your search into that area. For example, I just want to see how many occurrences are found within the country Nepal for this species. I can do a quick, I can make a quick polygon over the Nepal's boundary. I'm not being strict for sure here. You can be more strict, and the result will be shown accordingly. That's something possible. And it's also possible for us to it's also possible for us to provide the bounding coordinates for any area that we are concerned about, also provide WKT or geodesic files. That is, I think, mostly comes from GIS safe files. That's what can be done in location. So location is all about this. You can just search any specific locality in this section. And then comes the administrative areas, that is the global, based on the global administrative areas, and country or area. So the difference between these two is that in country or area, you can only search occurrences per countries. But in administrative areas, you can go to the lower level of countries as well, such as state, province, division, and so on. That's the difference between two. And let us quickly see how the number changes when I'm searching for, let's say, India, country or area, occurrences of Asian elephant there. It gives me 2,801 results in administrative area. And when I do the filter in country or area, it gives me almost more than 100 more results, right? 2,940. So this difference is because in for occurrences to be included under the administrative area, it should have the coordinates associated with it. But for country or area, if the publisher just provides the country field, Darwin Core country field as India, do not provide coordinates, will also be included. So that's the basic difference between these two, if you're using the same label country for search. And this is just a tip because we'll also be doing that in the exercise later on. So this is all about the filters that can be applied in occurrence that I showed quickly. Now let me go through individual tab here. In table, you can see the data that has been provided. You can choose which fields you want to show and which not. That's possible. In gallery, you can see the images associated with the occurrences. Only 4,773 has images, and you can click on the image and have a look at it. Map is the same as we saw in the species space. The occurrences with coordinates will be shown here. Yeah, one interesting thing here is we know that the Asian elephant is native to Asia, but we can see some points in Europe and also North America. So what's happening there, I will just let you know just later on. Taxonomy, because there's only one species, one taxonomy is shown, but if you just select more than one species, it will be shown accordingly. Metrics shows the summary of occurrences for you. There's a default way of showing where occurrences per month, per year, per basis of record, per license, per issues and flag, and per dataset is shown. And there is also a custom way by which you can summarize the occurrences. Let's say I want to see occurrences based on basis of record and country or area. Then it gives me a summary of occurrences in this table. So I wanted to show you the points of what's happening in North America. So let's look into United States of America. Most of the records there is from a preserved specimen from Natural History Museum. So it makes sense, right? It's also from material sample, maybe some part of an elephant or DNA or things like that. And then there's some interesting observation. It says human observation. Three occurrences says human observation, which should not be true because it's not naturally found here. Let's click on that three observation and go to table, which will show me only three occurrence. And when I click on the first one to see what's happening there, remember with each record, there are always issues associated if there is any. So you can also have a look on that. But I want to see where a human had observed this particular species. So I'll just directly go to the locality. And when I go to locality, I can see that it's from a zoo, but not actual natural observation. So it should ideally have been a living specimen, but they just provided it. So this is an error or issue with the record. So this kind of thing we need to be very careful when we are filtering or doing this search for the data set. So let's remove this filter for now also, and then download. You have to log in for download for sure. So I've already mentioned there are 4 types of download option for you. You can choose according to your need, and with the search refined searches, you can also always see the issues associated with the data you are about to download. So this is all about the occurrence and spatial filter. I will also quickly like to show you what interpreted data, when we talked about raw data and interpreted data, what does it mean quickly. When I go to publisher and choose India Biodiversity Portal, for instance, go to table and see the first record. If I move on and see, you can see the user has provided the date originally, and GBIF just interprets from here. It's 2024. It's year, month, and day, which is not very necessary field, but we can also see some more information here. The user have only provided original decimal latitude and longitude for the occurrence of that species, what GBIF has interpreted based on the latitude and longitude, saying that the occurrence is in India, country code is IN, and continent is Asia. So this is the difference basically between the interpreted and original data or raw data in GBIF. I will also quickly like to show you how a demo of the new website. You can also have a look on demo.gbif.org, although it's not live yet. So the homepage actually looks quite similar, right? But when we click on the occurrences, it looks drastically different. It's possible for us to just apply the filter in the headings here now, but we can also do individually here, like we did before. Elephas maximus and then apply. It's almost similar. We can apply the filter on your country or area. Something new on issues and flags that I wanted to show you earlier, and I also talked about it, is if I don't want this 29 record country invalid from my search, which has now 12,243, I can click on that and I can say exclude selected. So this will exclude my 29 results from the search. So that can be very useful for now. Location filter is almost the same. You have the map, you can draw and then see the occurrences within that, or you can provide geometry. You can also provide the bounding coordinates, so you can do that. And it's also possible to including coordinates or without coordinates. So this is about, and if it has any geospatial issues, you can also filter out that here now. There are many more filter options for you here. You can look into that. Obviously, we don't have time to go into detail on this one, but I'd like to quickly show you the dashboard. Let me delete now to show you how it's done. You can create your own summary of the occurrences. So let's say I want to view a table of occurrence. I want to view the map. Then I want to view basis of record, and I want to also view publisher. Yes, here. So I can decide what I want to see in the dashboard. I can choose accordingly, and this is very interactive as compared to the other one. So I will show you here. You can obviously change the way by which you visualize the refined search. You can do that. But if I quickly draw a polygon here in Nepal and try to, oh, it's beyond, but yeah, I'm just not being. If I just do that, this is very interactive. So when I try to filter results here, it will also change in table, basis of record, and publisher. So it will be very useful if we're doing certain combined, study with someone, search with someone, and we can share this dashboard with others as well. So this is a new feature that will be available in the new website. So this is quickly a view of a new website that is coming live soon, but now currently we have this website that we can use to filter out the occurrences from the occurrences tab. Yeah, so I will stop sharing my screen for now. This was a quick demo that I wanted to provide you. And based on this, we will also be doing an exercise. And I have some questions for you. Please go to gbif.org, go to occurrence, and then apply some filters based on the question that we have. I will share my another, I think I should have a presentation. Just give me a second. So this is the exercise that we are going to do. Uh, yes, you should go on gbif.org for sure. And based on what I've just shown you, can you please try searching for this occurrences, applying filters according to the question, and we can discuss later on the answers for each of these. Melissa, will we have like 10 minutes for the exercise?
Yes, we can have 10 minutes for it.
Okay. So we'll give you 10 minutes. You just quickly go to gbif.org and try to solve all of these, have answer for all of these questions, and we'll discuss it later on. Can you set the timer, Melissa?
Already said it. Do you want me to share the
I think we just keep a track because I have to see the questions. I have to show the questions as well.
okay.
Yeah.
Really, we are not able to see the questions. We are able to see your first slide.
Okay, let me try sharing again. Can you see now?
Yes,
Okay.
yes, we can
Yes.
see.
Okay. So maybe the time is up. Should we start discussing the questions and see if you have got the answers correct? I will also quickly show you how I would do the search in gbis.org at the end, but let us first see if you have the same answers or not. Can somebody, I mean, it's okay for you to just speak or type in chat. I'll stop sharing. The first question is, what are the total number of occurrences for Thailand? And I think we have already one answer there, right? Mm, Okay, we have different answer. Anita, did you apply the filter in like country or area, or did you do it in GADM filter?
Is Lily freeze?
Yes, looks like she has lost the connection.
Okay. Maybe we can just wait for a couple minutes for her.
You can actually continue if you have the presentation open.
I
And just go
don't.
on asking the
Yeah, but
questions.
we can try.
Yeah.
Let me check. Sorry, let me turn on my sheet.
Yes. Oh,
I'm so sorry, I had a power cut off, so
yeah, yeah, yeah. Continue. Please
disconnected.
continue. We
Yeah.
were trying to open the presentation to start.
Okay. So I don't know if you have answers for the first question, correct? Maybe. Oh, I can see chat. Yeah. Just let me open mine. Yes. So I have the result of eight. Um, 8277415. Yeah, most of us have the same answers. I was just trying to ask Anita if she why does she has a different one. When I'm doing later on, you can just figure out that. The second question: what percentage of the data do not allow for commercial use? Can you type in the answer? The number is also fine. Yes, 8.99. I think the number was 744,282, right? How many fossil records are there in Thailand? Fossil records for Thailand. Three nine two seven. Yes. How many records for the bee family Apidae are there in Thailand? For the bee family Apidae, how many records are there for Thailand? Correct. 17,648. Um, how many of the Apidae records in Thailand have an altitude above 900 meter? You have to apply the filter in elevation and see. 1922. Could not find that filter. Do not worry if you couldn't do that because I'll just be showing quickly. It won't take me much time. 1922 it is. How many of the resulting Apidae records with an altitude above 900 meter are georeferenced? Those that have coordinates. 1821, correct. And last question, how does the total number of occurrences change using GADM filter compared to one? Because one you were supposed to just filter out the country or area, and then when you use GADM filter, how does it differ? Like you can just give me the number when you're using GADM filter, because we already have a number when we're using country or area in the upper part. Mm, 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 mm. I don't know if Bharati is telling me the difference, but yeah. Can somebody provide me the answer for the last question? Why is
Please repeat the question.
How does the total number of occurrences change using GADM filter?
Okay.
Yeah, when you're using GADM filter as compared to country or area. Okay, I will just quickly show for those who do not know some of these might be helpful for those who have got correct answer. Great, but let me just quickly do it. You can see my occurrence filter search interface, right?
Yes.
So the total number of occurrence for country or area, Thailand. So I'll just search country or area here. It gives me this result, and most of us had the same answer. What percentage of the data do not allow for commercial use? Because I've talked about license, there are some records that do not allow for the use of data for commercial use. So you have to avoid CCBY-NC, and that is 744282. That becomes somewhat 8.99%. So that is the second question about how many fossil records are there in Thailand. So I explained about basis of record, and there is a section called fossil specimen that that tells us that the records are fossils. So 3,927. And then the next question was how many records for the bee family Apidae are there in Thailand? So in scientific name, we can just type in Apidae and not genus, but we want the answer for family. So if we apply the filter on family, then we have the occurrence to around Okay, it's taking some time. 17,648. Most of us had the same answer. So I think some of us has had trouble on doing this particular question. How many of the APD records in Thailand have an altitude above 900-meter? Because there's no filter that you can do in the simple filter. Please look into all filter, and you will find a section that says elevation, and then because it says above 900-meter, we can just select above and apply 900, starting from 900, and add, and it gives us result 1922. How many of the resulting APD records with an altitude above 900-meter are georeferenced? Whether it's georeferenced or not, it was in locality. If you just do including coordinates, it will give you the result. Sorry, elevation did not apply. I don't know why. I added it above 900. Add, and then in simple filters, including coordinates, right? 1821. So that was all the question was about. Let's remove these filters and see how it changes when we use the GADM filters. It was 8277417 when we are using country or area. Now when we write Thailand in administrative areas, then obviously it decreases because it only includes the occurrences that has coordinates. So yes, that was all the exercise was about. It's simple, but yeah, it might take some time for you to figure out what kind of filters is available and how you can do it. So just go on and explore gbif.org according to whatever research need you might have. Thank you. Over to you, Melissa.
Okay, and it's time for the group photo selfie and break. So please if you want to, please turn your camera on and we will have the group selfie. Okay, is everyone ready? Okay, look at the camera and 3, 2, 1, cheese. Let me check about the photo and we will share it after the workshop with the recordings. Okay, the photo is good. And because of time, I think we might shorten a bit for the break. So we will only take about 10 minutes break. Okay, and I'll also share the same thing, share the timer here. Okay. And okay, welcome back. Yeah, but I count I just saw someone said the break seems longer than before. But yeah, actually it's still 10 minutes. Okay, I hope everyone is back now. Please thumbs up or raise your hand or to let me know you are here now. Okay, good. Okay, then let's move on to the next one, the final section today. Okay, let me share my screen. Okay. Whoa, sorry. Okay, and let me go back. Oh. Okay, in this presentation, we will basically explore issues and flags of the filter features on gbif.org. So we will also provide related scripts for you to try in RStudio for tomorrow's session. Okay. And your GBIF download will not always you need to be aware of that. The download will not always be perfect for what you want to do with it. So there are a few things you should be aware of. Like during the indexation process over the raw data, GBIF adds issues and flags to records with common data quality problems. And this may contain useful information for you as a user to create fit-for-purpose datasets. When you try to download the GBIF mediated data, you might only see the appearance of datasets. So the deeper you dig or filter out, the better data for use. So there are some issues we will introduce what you need to notice here. Okay, the first is geospatial issues. When you search the occurrence on gbif.org page, you can see about the location here. There is a default geospatial issue button. And GBIF removes common geospatial issues by default if you choose to have data with a location. So for examples, zero coordinates, that which is coordinates are exactly 0, 0, which is a null island. And country coordinate mismatch is the coordinates fall outside of the given country's polygon. And the third, coordinate invalid. GBIF is unable to interpret the coordinates. And another, coordinate out of range. The coordinates are outside of the range for decimal latitude and longitude values. So, like this, 0, 0 coordinates will falls here, and which is the null island. Okay, nothing on the sea. And GBFlight also removes records that do not match their country code. But you need to notice that if your data location is about from the border of the countries, that sometimes might make some issues. So you can still include the records also, even they do not match with the country code. And the second issue is uncertain location. So this is an example that may need to be excluded in your analysis. This is a record from California, and the coordinate uncertainty in meters is about 12,205. So which is too large to know whether the species is on land or in the ocean or located somewhere. And often you will want to be sure that the coordinates given give a certain location and are not really 1,000 of kilometer away from where the organism was observed or collected. So we recommend not filtering out missing values, since publishers often leave them blank when the occurrence is fairly certain. And there are also a few fake values for coordinate uncertainty that you should be aware of. These values are errors produced by geocoding software and do not represent real uncertainty values. In the case of like 301, the uncertainty is often much, much greater than 301 and actually represents a country centroid. So country centroids are where the observation is pinned to the center of the country instead of being closer to where the animal, plant, or microbe was observed or recorded. And some publishers consider 0 and null to be equivalent, so that empty latitude and longitude fields for a record are given a 0 value. As a result, records end up being plotted along the equator and prime meridian lines. So this map like also shows like this. It's like you can see that it's easy to see the line at the equator. Okay. And country centroids, like what I mentioned previously, a country centroid is a point that is considered the center of the country, not defined by a polygon, and then we have a centroid attached to it. Why this occurs is when people are georeferencing what we often have when we have a specimen, as will I have typically textual information. So we may only have a country or a city or a text description for reference. So, for instance, we have the record saying this species was found in Japan. They will use the country centroid of Japan to put coordinates to that data, so that they may take the information on the label and produce coordinates based on country central, but not the place the species exactly found. So like okay, so the tips is to like use the distance from centroid in meters on a GBIF occurrence search to filter for country centroids. And the R package for tomorrow session, like coordinate cleaner, can also be used for identifying and filtering for country centroids. And greedy datasets. Graded datasets are a known problem at GBIF. Many like datasets have like equally spaced points in regular pattern like this. These datasets are like usually systematic national surveys or data taken from atlas. Georeference occurrence like are snapped to certain central point. Okay. So most publishers of like greedy datasets actually fill in one of the following columns. The first is coordinate uncertainty in meters, and the second coordinate precision and footprint WKT. Okay, this the last one will be found in Darwin Core archive downloads. And so filtering by these columns can be a good way to move gridded datasets. And for absence records, sometimes data publishers will include absence records where like they verify that a species is not present. And you can also know like if you know this is the habitat of your target species, if you also like download the absent records, you will know this species like will um appear in this habitat in which time, like in which season, which month, or at night or in during the daytime. Okay. but most of users like don't want these records. And actually, not many data publishers provide a current status, but GBIF like basically automatically interpret this from the individual count. So if the individual count is like above uh 0, that is like one, not it's not if it's not 0 and not blank, then it will turns to present data. So by default, both presence and absence records are shown when you search records on GBIF. And as absence records confirm that a species was not found at a specific locality when that area was surveyed, and this information can be useful in like what I mentioned. And if you are like only interested in presence records, you can filter for that by using the occurrence data filters like this here. Okay. And there are like other issues you have to filter yourself. The first field is basis of record. Okay. And it is like control vocabularies, and GBIF has like fossils and living specimens, usually a plant like inside the botanical garden, or sometimes an animal in a zoo. So you can use that. Even though this can be like a useful filter, data publishers do not always complete the basis of record field correctly, or there may be nuance in the data that may not be immediately obvious to a user. So you should like always double check your data before use. So for example, if you check like basis of record filter and filter the on human observation only, and when you go to the gallery tab, you can see the first one is not the obviously it's not the human observation. Okay, and it should be maybe machine observation. Okay. So always double check before you use it. And the second one is establishment means. In the Darwin Court term definitions is the statement about whether a Darwin Court-like organism has been introduced to a given place and time through the direct or indirect activity of modern humans. So is the process by which the biological individuals represented in the occurrence became established at the location. So it's also a controlled vocabulary terms. But unfortunately, it's not used very often. And you can see the filter here. There are some options in this term. Is the native, reintroduced, or native, indigenous, introduced, uncertain, or introduced. Another one, assisted like colonization, and vagrant. And the next is time duration is year, for example. Okay. So GBIF has many museum records that might be older than what is desired for some study. So you can go to the year filter and then set the duration you want. Okay, you can like set for the ancient data also. Okay. So now we are going to the final exercise. Sorry, it should be exercise 3. Okay. And it's like about issues and flags. And I hope you also already downloaded the exercise sheet. So you need to check like issues and flags from the family epi occurrences in Thailand. So it's like the filter from the previous exercise. Okay. And think about that. How would you use the flags to check the data? Okay, so here are some like questions and the steps you should do in this exercise. So the first is to view the metrics tab and review the occurrence issues and flags. And view the download tab here, like this one, and review the known issues. So there are two questions you should like think about in this exercise. The first is that would any of the issues cause you to do further filtering before downloading the data? And the next is which issues would be important for you to resolve after downloading the data. And then I'll also give you about, I think is 10 minutes also. Okay, I hope you already think about the answers. Uh, okay. Who wants to be the volunteer to answer the question? Actually, just like the simply answer about like which of the issues and flags you want to filter before you download the data or after resolve after you download the data and why. Okay, may I pick up somebody? Let me see. Mm, okay, Ashish, can you share with us? Ashish Dami? I hope I pronounced it correctly. Ashish, are you there?
He says in the chat he is not prepared his answer and working on it.
Oh, okay. Or maybe, sorry, I didn't see the, oh, okay, chat box. Uh, okay, Mizanur, can you share? Mizanur Ramen? Is that correctly? Hmm.
We have one person, Bharati, giving some answer in chat. Maybe we could
Okay.
score.
Yeah, Bharati, can you also share it with your microphone on mute? Bharati. Okay. Then I can see we can see. Okay. Yeah. See, Bharati's answer is like Okay, there are like like 40 invalid coordinates and coordinates is rounded. Yeah, but I would like someone to share, or you can type, all of you can type in the chat box, like which like data issue you will be like notice it when you deal with the data. Which issue is important for you to resolve it? And by the way, there the Dr. Nguyen also said like download is only supported for structured search, not for free text query. Yes, it's because it's downloaded from Darwin core terms and already like GBIF interpreted data. Okay. And okay. And Anita said before downloading to check whether all filters are correctly applied or not. Yes, you can also see all the filter you have on the download page. And Johnny said like recorded date invalid, recorded date mismatch, recorded date unlikely, and elevation mix minimum and maximum swept. Yes, these issues are really important. You need to notice that like recorded date invalid, that means the date is not, maybe the the date format is not correct or the date is incorrect. And recorded date mismatch is might be, and recorded date unlikely. There might be some like dates as out of range, which maybe is like future year of, or the wrong date in the like, maybe it's like let me think, 31 in February things. And okay, and elevation minimum and maximum swept. This issue you can still like download it, and then I think is also helpful and useful when you like just extract the range of the elevation, but you still need to notice about this. And coordinates rounded, Bharati said, actually it's not like really kind of the issues you need to resolve. It's just that means the coordinates, the number is like they provide the exact number and it's the rounded number. But you can also it's not the wrong. What I mean is that it's not the wrong value. Okay. And okay. Okay. So this index exercise, we just want to let you know, GBIF provide the issues and flags like here. You can see there are many issues and flags that can show you like that all the occurrence you download would like what kind of data issues they have. And like taxon ID not found is an issue, but you can still actually you can still download it and check the data if you can like still need, because they don't provide ID, but they may have like provide all other information that might be useful. Okay. And you can see like there are some the flags is not kind of issues, it's just a kind of notification. For example, like country derived from coordinates. It's like this data provider, some data publisher doesn't don't provide a country or country code. And then GBIF will like interpret it from the coordinates and give you this fields, like country field. So, and another is like, let me see. Okay. A current status inferred from individual count. That what I also mentioned in the presentation is that publisher often like don't provide the current status. So GBIF will interpret the data from the individual count, okay, to show its present data or absence data. And geodactic data invalid. If like some data publisher like only provide the decimal longitude and decimal latitude, sometimes they will like forget to provide the geodactic datum, but GBIF will assume is like WGS84, which is the common one from the Google Map. If you extract the coordinates from Google Map, then it's WGS84. Okay. Okay. So like what I suggest you to, if you like notice all the issues and flags from your download, if it's better to download it than resolve all the data issues yourself. And you don't like filter all the issues out before you download the data. Okay. VJ or Lily, if you want to add something on, you can just go it. And since it's
No, I
the
think I just want to highlight that we need to be aware of all these issues and be careful when we are using the data depending on what is your use case.
yes. Okay, and we still have 13 minutes to wrap up the session, but if you have any questions about all the section today, all the topics today, you can ask it now. Scientific Ashish just asked, scientific name ID not found, taxon ID not found. What does this mean? These issues and flags are related to Darwin core terms. So there will be the Darwin core terms is scientific name ID. If it's like refer from any checklist or backbone like GBIF backbone or catalog of life, you will have the scientific name ID of the species, and also taxon ID. But not every publisher will provide these fields in the dataset. But the data is still can like is like also can be used for analysis. So for me, I don't think this issues is the serious one. Okay. And what if there is a misidentified taxon? What do you mean? Do you mean the issue about like taxon match? Let me see.
Fuzzy.
That's fuzzy. Okay. You can still like download it and check the species name they provided, and then sometimes it's like this flag is how to say match with the GBIF backbone, and sometimes the backbone is not like always the real time updated to the latest species name. So you can still download it. If the species name is like the old one, then it will and not really match to GBIF backbone, then it will show as the taxon match fuzzy. but it doesn't mean you can still use this data, but the scientific name might be already like not the latest one, but you can still check with other checklists like catalog of life to see what's the latest scientific name of this species. Okay. Uh, is that, am I answering your question? Okay. And Anita said, asked which issues would be important for you to resolve after downloading the data. Okay, Vijay already said that it depends on your case, your use case. For example, location accuracy could be very important. For some, taxonomy ID could be crucial. And for me, like what Johnny said before, it's like the date, recorded date mismatch, recorded date invalid would be also important issues that I need to clean up for it. And then sometimes, not in this case, but sometimes, no, it happened often actually. The coordinate like mismatch or swap. Sometimes like people just like mismatch with latitude and longitude. Just these two fields just swap. So you still need to notice about this issue. Okay. Okay. And any other questions? And what I mentioned before, the country code mismatch with the coordinates. If that you see the issues and flag, you can still check if the coordinate is really mismatched with the country. That would sometimes happen between the borders of like two countries. Okay. If like no any other questions, let me remind you for tomorrow session, we will go for like API and R Studio for our GBIF package. Okay. And remember to install our studio. If you have like problem issues to like install it and run it properly, please like just stay here and raise your hand and Vijay will help you. Okay, Johnny asked, can we strictly go with coordinates instead of country code? Yes, you can like filter by like polygons and also provide like WKT footprint and also with coordinates. You have like many different options to filter the data you want. Okay. Uh, okay. And are there any issues when you install the RStudio? Please raise your hand. If you already installed the RStudio and run it like successfully, please raise your hand. Okay. So I think many of you already like downloaded successfully. Good. So tomorrow we will go. I hope the session will go smoothly. Okay, Vijay, do you want to add something on?
No, I think if people have our studio already running, then I don't have anything to share. Tomorrow we will actually use the R and R studio to learn how to query data from gbif.org in a different programmatic way.
Okay, be sure you already run the Art Studio and you can see the screen and the window that you can type in the script. That would be the definition of successfully run. Okay. Okay. Vijay, you're muted.
I can share the screen once and show what to expect.
Okay. And Ashish just asked, should we consider about the R or RGB version also?
Yes, you should have anything about just 1 minute. Yes. Give me one moment. Oops. My share screen just disappeared. Okay. Can you see my R Studio screen? Or not
Yes,
yet.
but not open yet.
Okay. Now you should be able to
Oh,
see it.
yeah, now is
Yeah. So as long as you are able to open something like this and see these four windows, that's all right. And as far as R Studio version, if you have anything above 4.3, that should be all right. If you have older than that, then you should uh, have a latest version installed. I have 4.5.2, but if you have 4.3 and above, that should be good.
Vijay, we cannot hear you well.
Oh,
Your
you cannot hear
voice
me well
will
again?
break. Yeah, can you say it
Is,
again?
is it better now?
Yes.
So what I was saying was we need version 4.3 or later. If you have latest, that's well and good. But if you have older than 4.3, then things may not run as expected. So make sure that you have that. Our studio version doesn't matter much.
Okay. If everything is good for now, then
Yeah.
just like saying goodbye to all of you and hope to see you tomorrow. Okay.